So our next subtopic is purification, और इसका हम पहला पार्ट डिस्कस करेंगे. Proteins, we all know that they are identified and characterized after the separation from the mixture, and this step depends on pH, ionic strength, dielectric constant or temperature of a solution, and their solubility as well. Separation of proteins, they also depends on the size of the proteins, on the charge present on the proteins, and heat, stability, etc., whether they are stable at high temperature or they are not, they are unstable. Effects of environmental factors, they are also important in characterization of proteins. And we all know that three-dimensional structure of proteins, they may be altered during separation step. So we will discuss here different methods which are used for purification of proteins. And first of all, we will discuss solubility-based methods. Separation proteins, of course, is based on their solubility in the aqueous solutions and may be done by following methods. And first of all, first method which we will discuss is salting out. During this method, as the name indicates, some neutral salt is added in the sample to precipitate the protein of interest, and proteins, they are precipitated when the salt concentration exceeds a critical value, which are then separated. So जैसे जैसे हम aqueous solution के अंदर प्रोटीन डिजोल्व करते हैं, सॉल्ट ऐड करते हैं, तो एक ख़ास कंसंट्रेशन के ऊपर जो हमारी प्रोटीन ऑफ इंटरेस्ट है वो प्रेसिपिटेट हो जाएगी. उसको हम सैम्पल करके उसके फर्दर कैरेक्टराइजेशन कर सकते हैं. मगर चूंकि इसमें सॉल्ट्स यूज़ किए जाते हैं, तो सॉल्ट जो होते हैं दे मे कंटामिनेट द प्रोटीन्स ऑफ इंटरेस्ट. Next, solubility-based method, that is known as isoelectric precipitation. What is isoelectric? Isoelectric point is the pH at which net charge on the protein is 0. And proteins, they precipitate at this pH while leaving other proteins in the solution. So, a khas pH के ऊपर, which depends on the type of proteins, जो protein होती हैं, precipitate हो जाती हैं, बाकी protein precipitate नहीं होती हैं, and proteins which of course separate, that can be separated. The proteins may have different isoelectric points which depend on their composition, and proteins which are stable at high temperature or at extreme pH may be easily separated by this method.